Hello, everyone. I'm very happy to be here for two reasons, really. It's the first time I've been able to give a public lecture since we've been struggling with the COVID issue. And also, it's the first time that I had the occasion to speak about SCIPIO and our solution for single-cell RNA sequencing. When I began thinking about single-cell sequencing, I was motivated by two thoughts. One was that in biology today, it's clear that the focus has to be on the cell. And the other thought was that to make single-cell sequencing really available for researchers all over the world in all sorts of conditions, it would be great to have a kit that didn't require an instrument and could be available anytime people wanted to do an experiment. So that's how things got started, and I'll tell you today how we developed this project. But first of all, I wanted to make a little introduction about cells, and I think everyone is familiar with the first principle of biology that was enunciated by Rudolf Virchow in Latin. Omnis cellula e cellula. He said that each cell derives from a pre-existing cell. Of course, we all know this. It's sort of a fact of life, but I think it's worth meditating on it a little bit. It's really quite awesome when we think about it, that all the diversity of biology, particularly with eukaryotic cells, probably came all from one event a billion years ago or so. And as we think about our own lives and the people in this room, we realize that each and every one of us came also from a single cell. So it's a perspective that we have to keep in mind. And an old friend of mine, Sidney Brenner, expressed it very well. He wanted to get out of the difficulty of deciding whether biology is totally reductionist or it's emergent. And he pointed out that if we choose the level of the cell, we avoid the question of whether our analysis should be top down or bottom up. And his approach is middle out, because from the vantage point of the cell, we can look down on the molecules that constitute it and look up at the organisms that contain it. So there's no doubt that the cell is important. And I'd like to introduce a second principle of biology. This comes from the observations that have been accumulating from single-cell RNA sequencing. This is one of the early key papers from Kosko and his colleagues who were studying the retinal cells. And you can see that they've made a nice clustering. And the more you go into the clustering at a higher magnification, the more you see that every spot, which represents a cell, can be separated from its neighbors. So the second principle of biology for me is that every cell is different. Of course, to make it sound serious, I put it into Latin, omnis cellulus differunt. So I'm not sure that this will become immortalized as much as everything comes from cell to cell, but it's an idea that we should keep in mind, and that may be perhaps the major take-home lesson of the talk today. But as we get in deeply into the subject, I put this little reminder to mention that, in fact, single-cell RNA sequencing has three different parts to it. And we can think that we might need three gras for each one representing one of the parts. Although, since I'm speaking to an English audience, we have to be a little bit more prudent. We can cover them up, and we can see that also they're now holding something that looks like a cell, and each one is holding a different colored cell. So the three pillars that we're talking about, I think, are pretty obvious to people who've been working in the field. We start at the left with a sample preparation, and that's the first step. Whatever we're working on, a biological sample of some kind, has to be put into the right form to carry out the analysis the sample preparation means somehow separating the cells and labeling them in such a way that we can go to the second stage, which is sequencing, and then into the third section of the operation, which is data analysis. And then finally, we sit and interpret our data. Now, many of the techniques that are available, including what we're doing at SCIPIO, really only deal with the first and the third stage. We leave the sequencing to the big companies that have produced all the wonderful sequencing machines. And our operation is able to work with whatever sequencing machines are available to the user. So really what we need to do is focus on the first and the third steps, and we'll start naturally with the first step. So how do we get the idea of making a kit? Well, actually, I went back to a trip I took to the Big Island in Hawaii, and I managed to climb to the top of the volcano, and it made a lasting impression on me. But what really struck me was the beauty and the structure of lava rock, and made me think all these little crevices and holes, if somehow they were filled with reagents and cells, that would be a way of doing single cell RNA sequencing. Of course, it would be terrible and impossible to fill these little holes, but thinking about it made me arrive at the idea that the way the lava rock is formed with the bubbles of gas that are eventually eliminated, that leave these traces, the same idea could be applied to cells in which the lava rock or a gel is formed around the cells. So that was basically the inspiration for trying to come up with a kit based on a hydrogel that would encapsulate the cells. So the idea was formulated, and patent experts helped me create a patent, and some investors thought it was a good idea. So it was a matter of setting up the company. I've decided why not do it in my favorite city, and Paris is a particularly favorable place for research these days because there's a great pool of talent, and the French government and the Paris region have quite a few incentives to help companies that are starting up. So we began in 2017 at a very nice location, the Paris Brain Institute, a relatively recent structure that has lots of services, and was very helpful to get us going. We started hiring people in early 2018, and by the end of the year, we were able to develop what we call the proof of concept, and I'll explain a little bit how that worked. And time goes on, and now we are in 2021, and we've actually terminated the design freeze for our kit, which we are planning to release for the market in 2022. So this was the team in 2019. It's grown considerably. This is the current team, over 30 people, quite a few more in wet lab research, but we now also have a substantial team in bioinformatics, and they've made huge progress, and I'll get to that a bit later, and our various management and communications people. So to get into the kit itself, as I said, we wanted something that would be easy to use on any benchtop for any researcher anywhere in the world. That should not require anything that's unusual for a laboratory, so it should be ease of use. There should be relatively no waiting time, shorter waiting time. Once the sample is obtained, even if it's in the middle of the night, you can immediately do the processing. No need to go to a platform. And all the steps are simple enough that no additional training is needed. So let's go into the individual steps of how it's done. So as I said, you always start with a biological sample. In our technique, the sample is put into a cell suspension and the typical barcode beads are added. The specificity of our system is that the barcode beads are connected to the cells directly by a chemical linker. Of course, the barcode beads in the usual way have poly-T tails, and since we know the messenger RNAs have 3' poly-A tails, the messages will end up on the barcodes, and that's the basis for the analysis. So in the next step, once we have the cells and the barcodes linked, we can dilute that into a hydrogel, a reversible hydrogel. So it's at the temperature where it's a liquid, a higher temperature. Then we can lower the temperature and the gel forms. And in that way, the cell bead pairs are isolated and held in place by the gel. However, the gel is not a total barrier to diffusion, and we can add the lysis buffer. So while the cell barcode beads are held in place by the gel, we can add the lysis buffer, and then the cells lyse and the messenger RNAs find their way to the barcode beads and poly-T endings. So the messages are connected to the beads. Then we can dissolve the gel, raise the temperature, the gel becomes liquid again. We can centrifuge the beads with their messenger RNA molecules attached, and then go on to the next steps of reverse transcriptase, amplification, and finally amplifying the cDNA to send it to the sequencer. So that's basically the process for step one. And to summarize the entire process, we made it lined up here. It's basically a day's work with 4 or 5 hours of hands-on time. We can apply this method to thousands of cells at a time, and all the operations only require standard laboratory equipment and no instrument. So then we have a material that is stable, so we don't necessarily have to go immediately to the sequencer. I'm sure everyone's had the experience of having to wait some time possibly to have a turn on the sequencer, but the material is stable at that point. And eventually we get the data from the sequencer, and then we have to go through the steps of analysis. As we began working with our own equipment and cells and our own collaborators, we discovered that, in fact, the day's work to produce an experimental result was straightforward, but depending on the subject, the data analysis could take many days, even weeks, and it could go almost on forever, because whatever you're looking at, you can always go into the subject more deeply. So that was where we decided that we also needed to put a significant effort into the data analysis part of our offering. And that's what I'll talk to you a little bit more about now. So the idea that we came up with was that the best way to have an accessible system was to use the cloud and have something that people can use and send their data to. So every sequencer is going to produce a FASTQ file. We can then analyze it for QC and go through the steps for analysis of the genes that are expressed, whatever we need, normalization, clustering, principal component analysis, and we get to the stage of clustering, and we can analyze individually which genes are expressed in which cells. So I think I skipped too fast there. Yeah, here we are. So the typical test of the quality of single cell sequencing is to do what's sometimes referred to as a barnyard plot. You take two cells from two different species, mix them up, and then do the analysis. And if everything works really well, in this case, we're using human cells and mouse cells. On barcode beads, we should find only human transcripts or mouse transcripts. No system is totally perfect, so you may find barcode beads with both human and mouse transcripts. And in this case, we can see the human transcripts in red and the mouse transcripts in blue, and the barcodes that have a mixture of transcripts are gray, and they more or less on a diagonal. So in this case, we don't see the high impact of overlapping barcodes on the blue and the red lines. But when we do a careful analysis, we can see that the percentage of multiplets, that is to say, barcodes, principally that barcode beads that have principally both human and mouse, is in the range of 3%. The other standard kinds of tests that we can do are looking at PBMC cells, and here we can do a typical clustering analysis, and we see that we've managed to separate the different cell types that are listed on the right very well. B cells here are quite far separated from monocytes, and we can see families of T cells where there is also quite good resolution. Ultimately, we wanted to compare our results with some of the other widely used methods now available in the world. Our institute also has a microfluidics based analysis system. So we compared on the same sample our approach and the microfluidics approach. And here we're presenting the results in terms of the percentage of cells in a PBMC experiment that are obtained by the two types of approach. And as you can see, the results are very, very comparable. There are, for some reason, a couple of neutrophils and basophils that showed up in the microfluidics experiment, but these are not generally considered to be part of the PBMC cell category. So we're very happy that we can meet any reasonable criteria for evaluating our approach, and we also were very eager to apply it to some interesting biological questions and experiments. And that was when we met Professor Bassam Hassan and his student Dania Shabani. We decided that would be a really exciting project to work on. So we selected their collaboration as the first collaboration outside of our own experimental group. And the project that they proposed had to do with Alzheimer's disease and part of the system of Alzheimer's disease that creates amyloid plaques. The plaques are produced by proteolysis of a bigger protein, the amyloid precursor protein, and that cleaves off what's the peptide that's called the beta amyloid peptide, and that is what is constituting the plaques themselves. So when they came to see us, they showed us some of their preliminary data, and we were terribly excited about it because it seemed like it was opening a window on a fundamental issue in Alzheimer's disease, and particularly the role of the amyloid precursor protein, which has never been really well established in humans. What they showed us was that when they used a a system of human cells where they could human stem cells, where they could knock out the amyloid precursor protein. They had 3 different lines in which the amyloid precursor protein was knocked out. They found that, and when they grew the cells in typical cell culture environment with the proper factors for neuronal differentiation, that in the knockouts, one of the markers that is normally a sign of a neuronal differentiation, the marker disappeared very quickly. And on the other hand, another marker that was a sign of the later stages of the neuronal differentiation appeared quite early. So basically, the appearance of these markers indicated that when the amyloid precursor protein is knocked out, the differentiation is actually moving ahead more rapidly. So it's as if the amyloid precursor protein is serving as a kind of brake, and for some reason, when the brake is not on, the differentiation goes faster, and in the long run may lead to production of the pathological condition of Alzheimer's disease. So we were provided with samples of the knockouts and the controls, and we found a clustering patterns that gave us information on the various cell types. And in addition, we found that there were a particular cluster of neurogenic precursor proteins that were only present in the knockout, which confirmed that the knockout was liberating expression of these particular genes. We could also do a pseudotime analysis, and this confirmed that the knockout had a different timing in the appearance of some of the critical markers. So this work has been submitted for publication, and the bioarchives are holding the latest version of the paper, and 2 of our colleagues, Azad Safarian, and did the biotechnology, the bioinformatics work, and Jun Komatsu performed the experiments. So in this brief presentation, I hope I've convinced you that we can achieve single-cell RNA sequencing on a large scale without instruments, merely by using a reversible hydrogel, that our system is suitable for serious biological discoveries, as we've just shown with the Alzheimer project. And we're delighted to imagine that the kit will soon be available for use by people everywhere in early 2022. So it's important to acknowledge the people that helped us get to this stage, our various investors, and the help we got from our hosts in the Paris Brain Institute, and some additional funding from sources in the Paris region and the Ile-de-France. So thank you for your attention. We think we're on the verge of even a greater expansion in the field of single-cell RNA, and we're also following up all the work that's going on in various approaches to single-cell RNA with our newsletter, and this is available on our website. And to finish, I'll just remind everyone that we're expecting a huge eruption in more and more data of single-cell RNA, and we can go back to our Volcano illustration, and this time this is a big eruption of data. Thank you for your attention.